Hello, and thank you for joining my presentation on clustering adverse events of COVID-19 vaccines across the United States. Quick background on myself. I'm Ahmed Askar, a PhD candidate at George Mason University. I'm also currently at the U.S. Food and Drug Administration as a geographer. I would like to add that this work was solely done for my PhD studies. Quick disclaimer that the opinion here does not reflect my employer. The agenda is as follows. I'll start with the motivation and the purpose of my research. Then I will give a detailed overview of my research. After that, we'll take questions and possible feedback. On the motivation, discovering all side effects during the vaccine and drug development process is impossible. Adverse side effects of vaccines and drug may vary over space and time due to different population factors such as genetics, language barriers, environmental factors, which might be surrounding the, the drug intake, and the drug or the vaccine quality from the manufacturing process or the uh, or possibly on the supply chain, and including also the care that you get from your healthcare professionals. I listed a few examples, a few example factors in the next slide. However, there are many unknowns, unknown variables and factors. I'll expand on these, which is going to tie into the why of my research. During the vaccine and the drug development process, the approval process, the potential harm and the benefit of the drug is weighted. And as you know, the clinical trials are getting less diverse, then it's very hard to see the full picture. Discovering all side effects is normally very difficult. We all know that human beings are so similar yet so different. Take two twins, for example, they might be similar in some aspects and different in others. This sometimes is reflective of how adverse events present themselves in the general public. The clinical trials are massive projects, and sometimes it's impossible to get a sample size that is truly a reflective of the population. An example of a adverse event which might hit certain demographic is the effectiveness of albuterol, the asthma inhaler. It's less effective in African American and Puerto Rican children compared with Caucasian American and Mexican children. Another drug, abacavir, is an HIV drug which can cause life-threatening adverse event if a particular gene HLA-B5701 is present. Prevalence rates are higher in Caucasians as compared to other demographic. On the flip side, the same genotype makes you resistant to malaria. Language barriers and a not good understanding of the hospital discharge instructions can lead to sometimes to an adverse event. This is a quick overview of the environmental factors which can influence the effectiveness of the drug or might even cause an adverse event. Environment here means that the environment of the human body. These are not an exhaustive list, however, they are some examples. Drug to food interaction, drug to disease interaction, and the common one that we normally hear about, the drug to drug interaction. Another way that adverse events may arise, and normally this makes the headlines, is the issue of the drug quality. This was an event I worked on several years ago at the FDA. In 2012, 753 patients in 20 states were diagnosed with a fungal infection after receiving a steroid injection manufactured by a New England compounding pharmacy. Uh, they, they were receiving it for back pain, and this resulted in many patients being hospitalized and dying of a fungal infection due to unsanitary manufacturing process by the compounding pharmacy. And during the FDA investigation, an investigator described it as a fungal zoo during his investigation. The outbreak was the largest public health crisis ever caused by contaminated pharmaceutical um, drug. So, as I said before in my previous slides, I want to identify an adverse event, adverse events associated with an FDA approved drugs or vaccines, and then identify any special patterns. Some questions that I might ask while doing research is, are adverse events constant for each special region, or are they not? And just the adverse events repeats across space and time. Some data characteristics, the various the vaccine adverse events reporting system, VAERS for short, is a national vaccine safety surveillance program overseen by CDC and FDA. It contains post-market adverse event reports. And FDA and CDC scientists look for unusually high numbers of reports of an adverse events after a particular vaccine or a new pad or after a particular vaccine or a new pattern of an adverse events. If scientists see either of these situations, they go then into a full focused studies in other systems, and these are done to determine if the adverse events is or is not the side effects of the vaccine, or yeah. So a quick overview. So I say a particular person receives a vaccine, and then you may or may not have an adverse event. If you do, then um, the um, the adverse events might be a true adverse event or a just a coincidental health event, not related to the vaccination. Um, but either way, the report is submitted to VAERS. VAERS, they are healthcare professionals or patients or parents or caregivers or vaccine manufacturers can all report to of VAERS for a adverse events, and then the report is processed and done some MedRAC coding, and also done some data entry and quality control, and then after that, follow up. There could be some follow up to obtain health records, and after that, the data is transmitted to the CDC and FDA for further look. This is a sample various data. You will see that there's an adverse event ID, the vaccine or the drug, and the location that it was administered, and the set of adverse event that this particular this particular event, ID, what adverse event they received. One thing I would say about FAIRS is that it is a public database, so due to the HIPAA regulations, data is summarized at the state level. So the data run was done for the 3 COVID-19 vaccines, Moderna, Pfizer, and Johnson & Johnson. The temporal study for this research was for 2021 from January to June. The study area was the United States. So you might ask yourself, why why COVID-19 vaccines? Of course, for researchers, it's great when your current research is of great interest to the masses. This brings, of course, both good and bad. Good as in there's plenty of data from different kinds, from different credible sources, and the good another good side effects is that journals are interested in this research, and there are many others, so etc. etc. The bad, of course, there are a lot of misinformation. So research is needed to quell any misinformation with evidence-based research. So research or further research is needed in this aspect. So why temporal? Why did I choose the temporal selection and the study area? It was just due to the data availability. So possible solutions that will drive my research will be both geospatial science and machine learning related. So the roadmap that I would take is that first the collection, the pre-processing is going to happen, and then after that, topic modeling, and after that we'll do some similarity search, and after that we'll do spatial clusters analysis and evaluation. So due to the time constraint, I will not go into the collection, the pre-processing. I would start with the topic modeling. So for the topic modeling, we used LDA. LDA is an unsupervised learning. It's an efficient method for discovering the hidden topics in a document. The output are document classified into the discovered themes and topics. We tried using frequent item sets, but there's some limitations. Of course, there's a limitations that it runs too long, so computing resources, and the other limitation is that it doesn't handle well the semantic understanding of an adverse events. So it wouldn't know between soda versus pop, or balance disorder versus fall versus balance problem in the database. So we used LDA. So how does it work? First, we want to find the topic in the document. For our research, adverse event will be the words of a document. Documents will be the spatial regions. So once the words from the document are extracted, the algorithm groups these words into topics. Finding the optimum number of topics was challenging. So we have to balance between maximum information retrieval and also minimizing the noise. So the number of topics selected, we used using coherence scores, and we settled with 10 topics. So can we find similarity between space? Of course, if you look from the aspect that you can compare document A to document N, so similarly we can also compare region A to region N. So can we find similar topics across space? I think we can using distances. However, we have to plot them into a multidimensional space, so it's easier to understand our method and process. Each spatial region might have some topic more dominant than others. We'll plot these. If we plot these, we will find the distances between them. The closer that they are to each other, the more similar. So for example, here we have a 3-dimensional space, but if we have more, we can easily use similar steps. And next, we do have an array of distance algorithm to use, but however, we stick with Euclidean distances for simplicity. We can just plug in the topic distribution per region and then calculate the distances between each region. So here, once we find the similarity distances of our latent features between region using the Euclidean distances, we'll move to the clustering. So next, we use the hierarchical clustering. It treats each observation as a spatial cluster. When doing the bottom-up approach, the advantages of hierarchical clustering is its ease of handling any form of similarity or distances. The result here is a result of a pairwise similarity matrix from a latent topic using Euclidean distances of COVID-19 vaccines. And then we use the hierarchical clustering analysis on top and the side of the image. The darker color corresponds to the higher pairwise similarity. We observe a large group of mutually similar states having smaller nested clusters of similar states, thus explaining why we chose hierarchical clustering. Now that we have our clusters, we want to find if these clusters have any special patterns. Here we'll use both global and local local run to test any spatial autocorrelation test. So if you go back to the slide to understand how I use the probability of latent topics to further study their special patterns. Here, each US state in our study had its latent topic probability from topic 1 to 10, and then we use these probability to run the Moran test. Moran I test, which is a test for spatial autocorrelation test. Again, Moran I is a test statistic to test the hypothesis that a spatial phenomenon appears uniformly at random without any spatial patterns, or the opposite. So moving to the results. So going back with each state having its corresponding probability for each topic, we use global Moran to measure the spatial autocorrelation, and we observed in table 3 of our paper that out of the 10 topics, 6 topics show no spatial autocorrelation, which means that we were unable to reject the null hypothesis of a random pattern. And one topic showed negative spatial autocorrelation, implying that significant dispersed pattern. And 3 topics exhibit positive spatial autocorrelation, meaning that spatial clustered pattern. For the clustered pattern of topic 1, 8, and 10, we observed highly significant p-values showing that these 3 topics of COVID-19 adverse events do exhibit significant spatial autocorrelation. This result showed that some latent topics among the adverse events, among the adverse effects of the COVID-19 vaccines, indeed depend on location. For a deeper study, we show that the local indicator of spatial autocorrelation, or LISA for short, for topic 1, 8, and 10, showing that the spatial location of clusters of regions that exhibit high or low probability of each corresponding topic. We also observed the high to low, the outliers, a region having high and low topic probability that are surrounded by regions having low to high topic probability. These significant clusters show some adverse effects indeed vary locally. Uh, this warrants a further study to understand why certain regions of the United States exhibit different topics of adverse events. So, in conclusion, we tackled the problem of measuring similarity and dissimilarity between adverse events of COVID-19 vaccines observed in different regions. Our measure, we leverage a topic modeling approach using LDA to map each adverse event from a textual set of adverse effects to a latent topic distribution. Our results show that some latent topics of COVID-19 vaccines shows significant positive spatial articulation, and our local analysis also of spatial articulation showed that this certain topic of adverse events have increased or decreased likelihood in different parts of the United States. And we hope that to speed the time in identification and prioritization of interesting or potential adverse effects of concern for further analysis. In return, we believe that this research is going to improve the surveillance model in post-marketing phase of pharmaceutical drugs and vaccines. And for future work, we will look at the difference of macro versus micro spatial analysis, and also same for the temporal analysis. So finally, our work was all done in Python. All the analysis and the data management, all the data and process was open source, and you can find our notebooks and our project on GitHub on the website provided here. And finally, questions. Thank you.